Welcome to the program. I'm your host, Neil Howard, here on Health Professional Radio. Thank you so much for joining us again. Going to be having a conversation with Dr. Carolyn Loew this morning. She's president and CEO of Glympse Bio. It's a clinical stage biotechnology company. And she's joining us here on the program this morning to talk about the company's development of tunable biosensors that are capable of non-invasive disease detection and monitoring. Welcome to Health Professional Radio, Dr. Carolyn Loew.
Thank you, Neil. Delighted to be here today. Really welcome the opportunity to speak to you.
Well, give our listeners a bit of your background, a look into your professional, your area of expertise, and then let's talk about what biosensors actually are.
Absolutely. So I have over 20 years in the pharmaceutical industry, predominantly in drug development. And I joined Glympse as CEO a little over 2 years ago, and we've been building the company into the clinical stage organization that it is today.
Tunable biosensors. What are biosensors and how are they tunable?
You know, it's a great question. So at Glympse, our mission is really to change the way diseases are tracked and treated. And we're doing that with, what we call our tunable, non-invasive synthetic biomarker technology. And this is something that can really have very broad patient and disease impact. It's something that can both detect disease as well as monitor disease progression. And if you sort of think about, probably your experience personally, maybe of friends and colleagues, but also probably the experience of your listeners in how they manage their patients or even their personal experience. This whole question of how disease gets, how you identify whether you have a specific disease and then how that disease gets treated, how you determine whether the disease is actually responding to treatment is, a perennially difficult question. And there are actually many difficult diseases today that we just do not have good tools to do this for. And that's what our technology is aimed at addressing. And the technology itself is very simple in how it works. It can detect real-time changes in disease using sensors that are bioengineered and tunable to pretty much any disease. And so what we built and why we're so excited is essentially this broad biosensor technology that's set to redefine both the diagnosis of disease and tracking the treatment response.
When I'm thinking of disease detection, I'm thinking of a blood test. I'm thinking of a biopsy. Are you talking about replacing biopsies? Is this something that is implanted in the body that constantly monitors the changes? How exactly does it work to detect certain diseases?
Yeah, and so this very much depends on the disease. So as I mentioned, the tunable sensors are designed for each disease state, and how they work in their current form, and this is something that will develop over time and will change depending on the specific disease. But in the current form that we're developing for NASH, which is our first indication, they're injected into the body, and there they localize to the disease tissue, in this case, the liver. And they measure the activity of proteases that are dysregulated in the disease. And then within an hour of administration, the sensors are excreted in the urine. So it's a simple urine collection. They're detected using standard laboratory approaches. And then it can be analyzed with what we have a proprietary classifier that's very unique to the disease state. And then they're reported as, this is very important, as clinically actionable results. And then what we actually do, which is particularly interesting, I think we can, for each patient, we can amass that data into a data engine, and that enhances the precision of the classifiers and it allows us to power future discovery. And so there's sort of this continuous learning process that we go through. We can not only report specific patient actionable data, but we can continue to learn from the patient data in an aggregate form, which allows us to learn more about the disease, and continue to eventually deepen, physician information on how to diagnose and treat diseases. And NASH, as I mentioned, is our first indication. And in the case of NASH, we would actually be replacing a biopsy, which is a very painful procedure.
Mm-hmm. How long do these biosensors continue to monitor once they are introduced into the body?
Yeah, so it's a point in time measurement. So we administer the sensors and we get a report about an hour later. So to get repeat measurements, you have to repeat the administration of the sensors. And generally, how we think about it right now is that that's something that you would want to repeat, sort of serially as disease is progressing or hopefully regressing for a patient. And so we think about this in two different ways. Firstly, for diagnosis of disease. So, a patient comes to a physician's office. We're trying to understand whether they have a disease and the severity of that disease. And we can tell that spectrum. So that's sort of the first thing. And then second thing is the patient goes on to treatment. Can we determine whether that treatment is working? And so over time, the patient with the physician, the patient can use this diagnostic to determine their response to treatment. And so, that might be that it's being used depending on the disease. NASH is a very slow progressing disease, so you might want to only use the diagnostic every 3 or 6 months. Mm-hmm. In cancer, you might want to use it much more frequently. But the idea would be that you would use it, serially over time to determine response. And what's been very interesting is in our animal studies to date, which we've compared to comparable clinical measures, we've been able to see response to treatment far earlier than histopathological changes. Mm-hmm. And so that's very encouraging for us. What it means is that today patients are waiting to see changes in response to treatment using standard diagnostics, and it's taking a very long time. And it appears that our diagnostics, because we're measuring direct biological response with proteases, we potentially will be able to see response to treatment far earlier than you can today. And that's good news for patients because if their treatment is working, they know to continue. They're getting very positive feedback. But if the treatment isn't working, we'll know early and we'll be able to revert them onto another treatment, which is very important, particularly in diseases like cancer that are progressing quickly.
Is there a total lack of any adverse effects when the body detects the biosensor?
Yes, great question. So far we've completed our phase 1 clinical studies, and we saw no safety signals, and in all of our preclinical studies we had an extremely clean safety profile for the sensors, and so the great advantage of this appears to be that we're detecting the activity of the disease without any sort of biological effect on the body, if you want. So there doesn't appear to be any, thus far at least, we're not seeing any adverse effects on the body, which is particularly great news for this kind of intervention.
You mentioned several conditions that the technology would be effective with, NASH being one of them. Were there other indications that you were pretty sure or positive that this would be effective in?
The technology is based on protease-mediated disease, and there are many protease-mediated diseases. Mm-hmm. So we're looking broadly at fibrotic diseases. NASH is the leading indication, but we're also looking at oncology. The fundamental the single driving mechanism in oncology of T-cell response is protease-mediated. Infectious disease are highly protease-mediated diseases, and we're looking at those as well. And there are sort of over 20 major diseases that are protease-mediated. So there are many areas for us to explore, and we're very excited about that potential. The beauty of this platform is its tunability and the fact that it anchors to protease disease mediation.
Where can our listeners go online and get some more information about Glympse Bio?
We have a website, www.glympsebio.com, and we certainly, there's a lot of good information there about the company, our technology, and where we are in our development. We're also on social media, Twitter and LinkedIn, and we'd encourage your listeners to follow us there.
Well, I appreciate you joining us here on the program this morning, Dr. Loew. Thank you so much.
Thank you for your time, and thanks to your listeners for tuning in today.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. Carolyn Loew. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes. Listen in, download a SoundCloud, and be sure to subscribe to our YouTube channel at youtube.com, Health Professional Radio.